Alright, welcome back to another MCAT prep video, and today we're once again going to be continuing the biochem section by talking about protein separation techniques, the isoelectric point, and gel electrophoresis. And so a few learning objectives that you should meet by the end of today's video are that you should be able to understand and calculate the isoelectric point, also known as PI, be able to understand how electrophoresis separates proteins, and be able to compare common protein purification methods. And so before we get started, one thing I just want to mention real quick is that the whole premise behind today's video and what you should be thinking about as you watch the video is how we are able as scientists to take advantage of the physical properties of proteins in order to separate them. So moving into content, the first thing we're going to be talking about is the isoelectric point, also known as PI. And what this is it's essentially the pH at which a molecule has no net electrical charge. And so even though the molecule itself has no net electrical charge, it is still possible that it can have within it positive and negative charges, but they just have to cancel out at the isoelectric point. And so you might be wondering, why is it that charges change, or how do charges change? How is it possible that a molecule with different charges can have an overall charge of 0? And so the reason this is possible is due to side chains and different positively and negatively charged groups like the carboxyl group or the amino group like I've drawn here on this lysine. And so as you may remember when we first talked about amino acids, each of these groups has a different pKa value. And so when the pH of the solution that these proteins are in changes relative to the pKa, these groups can either be deprotonated or protonated, which will affect the overall charge of the molecule. And so I think this will make more sense when we look at an example of how to calculate PI, because that really helps show you what PI actually is. And so let's do that. So one thing you should always be doing when you're calculating PI or starting to calculate PI is just draw a number line. And I've found this is really helpful to understand what PI really is. And so on this number line, what you're going to do is you're going to put down the pKa of every group within the molecule, peptide, or whatever you're trying to find the PI of down onto the number line. And so in this case, I've drawn a lysine here. And so we know that the amino group has a pKa around 9. The carboxyl group has a pKa around 2. And the R group of lysine has a pKa around 10.5. And so what you're going to do is you're just going to draw these pKa's onto the number line. So we have 2, we have 9, and we have 10.5. And so now that we've drawn all these pKa's down to our number line, we're going to try to start thinking about when the net electrical charge of the lysine is 0. And so let's just think about that. Let's think about when it is possible that the net electrical charge of our lysine is 0. Well, we know that we have a pKa here around 2, a pKa here around 9, and a pKa here around 10.5. And that when our pH of our solution that the lysine is in is going from one side of this pKa to the other side of this pKa, there's either going to be a deprotonation event or a protonation event. And we can see that through following these protonation or deprotonation events in a logical or stepwise manner, we'll eventually be able to see where the overall charge of the molecule is 0. And so let's do that. So let's just assume that we're starting down here with a pH that's less than 2. And we're going to end up here with a pH that's greater than 12.5. So let's do that. So let's say we're right now at a pH of 1. So this pH value is less than every single pKa value of every ionizable group on our lysine. And so what's going to happen? Well, we know that if pH is less than pKa, we're going to have groups that are protonated. And so remember, you can think of pKa as the pH at which a group is going to get rid of its protons. And so if the pH is less than that pKa value, these groups are going to be holding on to their protons. And so let's see what happens. So if every group is holding on to their protons, we're going to have a proton here, we're going to have a proton here, which is already drawn, and we're going to have one more proton down here. And so what's the overall charge of our molecule? Well, let's take a look. This group has a charge of 0. This group has a charge of +1, and this group has a charge of +1. So overall, at a pH of 1, our charge would be +2. So now let's keep going. So we've thought about a pH that's less than 2. So now let's go to a pH that's greater than 2, but in between 2 and 9. So let's say, for example, we just pick a pH equal to 5. So what's going to happen? Well, now we're crossing over the pKa of 2. So the only thing we should expect is something to happen to the group with a pKa of 2. And what's going to happen? Well, now if our pH is greater than our pKa, so our pH is greater than our pKa, we're going to expect a deprotonation event, right? Because this group, this carboxyl group, wants to give away its hydrogen at a pKa or a pH of 2. And so now if the pH is all the way at 5, it already threw that hydrogen away, so it's gone. And so now what we're going to have here is we're going to have a negative charge. And so let's count again. What's the overall charge of our protein? So we have a -1 here. We still have the +1 here, and we still have the +1 here. So this plus and this minus are going to cancel out, and we're left overall with just +1. So let's write that down. All right, and so now let's keep going. So what's a pH in between 9 and 10.5? Well, let's say it's 9.5. What's going to happen? So again, we're crossing over another pKa boundary. And so we're expecting the group with a pKa of 9 to now get deprotonated. And that's exactly what's going to happen. So one of these hydrogens in this amino group is going to get lost. And overall, the charge of this group is now going to become 0. So let's count. What's the overall charge of our protein? So we have a -1 here, a 0 here, and a +1 here. And so these cancel out, and the overall charge of our protein is 0. All right, and then, based on the following pattern, you can see that if we went somewhere over here, and we, like, for example, went to a pH of 12, the overall charge of our protein would now be -1, because this group would get deprotonated, and now all we're left with is just this negative charge over here. And so now that we have a clearer picture of the charge of the lysine at different pH points, we know that the pI is going to be somewhere in the middle here. So somewhere in between the pKa of 9 and somewhere in between the pKa of 10.5. And so that's exactly what you do. You find where the protein or molecule has a net charge of 0, where it is a Zwitter ion. And then you average the pKa to the left and the pKa to the right. And so the pKa of the lysine would be, the pI of the lysine would be equal to 9 plus 10.5 over 2, which equals roughly 9.75. And so that's how you find the isoelectric point pI of a single amino acid. But keep in mind that this method also works if you strung multiple amino acids together. And instead of just looking at the carboxyl group and the amino group, you would have to look at the R groups of every amino acid that can be protonated or deprotonated in the peptide. But remember, you don't have to think about each carboxyl or amino group because there's only going to be one of each in a long peptide. And so if you think back to what I said at the beginning of this video about how we're going to be using peptide or protein properties to separate them in solution, you might be wondering, okay, so why is this pI value so important? And it's so important because when the proteins are charged, or when they are not charged, and that is they are at their pI point, they have a net charge of 0, that really affects their solubility. And so you can take advantage of this property of proteins, or even bigger peptides, and play with the solubility and the pH of the solution in order to separate them, which is something we actually will see later. And so the next thing we're going to be talking about is the relationship between pH and charge. And so the way I want you guys to think about this is sort of like a tug of war between team positive and team negative. And so you guys might have been able to see from the previous slide how depending on changes in pH of the solution, our charge on our protein is actually going to change when the pH crosses those pKa thresholds. And so for example, if our pH is less than our pI, which is what we're seeing here, remember that every time the pH was less than the pI, which is when the pI is when the protein has a net charge of 0, we're going to see a positively charged protein. And so if you think about this in terms of the tug of war, well, that means that the team positive is winning. There's so many free hydrogen ions in solution that all those hydrogen ions are so crowded that they're binding to the groups that can be protonated on the protein and creating a positively charged protein. Now let's think about pH equals pI. So this is when our protein is going to have a net charge of 0. So we're not going to have a charge on our protein. And so, in terms of our tug of war, we can think about this as the proteins being right in the middle. And there's a perfect balance between the groups that can be protonated and deprotonated and the solution. And so our protein is not going to be charged. And so now let's think about pH is greater than pI. So now we know that the solution is very basic. There's not many hydrogen ions floating around. And so all the hydrogen ions on our protein are going to be going into solution. And we're going to be left with a negatively charged protein. And so in terms, again, of our tug of war, now team negative is winning. Our protein is now negatively charged. And so these relationships are important to understand because you may be given questions where you're told the pH of the solution is this and the pI of your protein is this. Do you expect a negatively charged, positively charged, or protein with 0 charge? And so it's important that you just quickly can be able to answer those questions based on this relationship. All right, so now let's move into the real main point behind this video. So now we know how proteins can be positively charged, we know how they can be negatively charged, and we know how this can be affected by the pH of our solution. And so why is this important? Well, this is why. We can use techniques in the lab to separate proteins based on the physical properties or the charges we expect them to have. And this is hugely important all across science because if you can separate proteins, you can isolate them and you can study their function much easier. I mean, think about how hard it is to study the function of one protein in cells with hundreds of thousands of protein. It's almost impossible. How do you know that the protein you're interested in is doing what you see happening when there's hundreds of thousands of other proteins in that same environment? And so that's why it's so important that we can isolate proteins and we can do that in a consistent way. And so let's talk about one of those methods. So we're going to be talking about electrophoresis. And here on the screen, I have a diagram of, a simple electrophoresis study. And so let's just make sure that we're on the same page here. So a few components, or a few of the more important components in this setup to understand are the gel membrane. And so usually it's just called a gel. We have wells in which our sample, in this case, which is green, is being inserted into. We have some sort of a current being applied. So you see at the top there's a negative charge, and at the bottom there's a positive charge. So that means we have a current going through the membrane. And although it's hard to see, there is a buffer or some sort of liquid that's surrounding the membrane. And then again, just to help facilitate the creation of that current, there are these electrodes. One up here, negatively charged, and one down here, positively charged. And so now let's think about what this all means. Well, we know that proteins can be positively charged or negatively charged based on, their PI points and based on the pH of solution. And so if you know what a protein's PI is and you know the pH of your solution, you can use this charge, you can utilize this current to help proteins migrate through solution. And so let's say you had 10 proteins, all the same size, but different charges. Those proteins that are more negatively charged are going to end up further down this gel when you insert them in here because they're being repelled by the negative charge and attracted by the positive charge. And so these proteins are going to migrate further down the gel. But in principle, in reality, we're not just separating proteins strictly based on charge usually. We're separating proteins based on charge, based on shape, and based on size. And so you might be wondering, okay, well, I understand the charge. I see how there's a negative charge. I see how there's a positive charge. That makes sense. But these two can be a little confusing. How are we separating proteins also at the same time based on their shape and size? Well, this is how. Here's a picture of what a typical agarose gel membrane looks like really up close. And so now you might see in this picture how there are certain holes that are bigger, there are certain holes that are smaller, but overall this gel can be seen like a mesh, like almost like a spider web, a 3D spider web that's sort of growing in every direction. And so bigger proteins are going to have a harder time fitting through this mesh. So even though they may have a charge that's forcing them down the gel membrane, they're going to be getting stuck in these holes. And so they're going to migrate more slowly. And so now you can see why proteins with larger size, larger shape, are going to end up being higher on gels. And proteins with smaller size, and they're able to navigate through these large holes, are going to end up lower on gels. And so electrophoresis utilizes charges, it utilizes shape, and it utilizes size of proteins to separate them based on these physical properties. So let's dive into that a little bit more. So we know that we can separate proteins based on physical properties, but you might be wondering, okay, what if you just want to separate on charge? Or what if you just want to separate by shape or size? What do you do? Well, that's a good question. So the technique we looked at before, or the one that I was explaining before, is what's known as a native page. And so the most important part of a native page is that it maintains the native protein structure. And so size and shape are going to be a factor as well as charge when it comes to protein separation. But because you're maintaining the protein size, shape, charge, You're essentially keeping your protein exactly how you took it out of the cell. And so, why this technique is important is because you're also going to be maintaining enzymatic activity. And so it may be possible that, your protein is separated with other proteins of similar size, similar shape, similar charge. But then you can add substrates onto your gel, for example, that are radio labeled, and only the specific bands with your protein of interest are going to have a reaction. And so you can see, okay, that's where my protein is, that's what I can isolate. And so this is really like your generic case of electrophoresis, your native page. You're not really manipulating the proteins in any way, but you're able to find out, what's their general size and, even might be able to infer what their general charge is. And so this method can be used if you really have not too much information about your protein. You might know what substrate it reacts with, and you just want to get a feeling for maybe the size of the protein. That's when you would use a native page. In addition, if you want to get a little bit more specific, now is when we're going to start talking about the SDS page, for example. And so what does this do? Well, the SDS page essentially works by taking your protein and then you unfold it. So you use detergents or denaturants to unfold the protein. And then after you unfold it, you coat it with a uniform negative charge. And so what does this do? Well, if you think about the three primary components that we're looking at in electrophoresis or properties, size, shape, and charge, essentially you're knocking off charge. Now that's not a factor anymore because our proteins are unfolded and they have a uniform negative charge. So it doesn't matter what proteins you put in here, they're all going to be negatively charged. And so now an SDS page is a way that you can filter or separate proteins exclusively by size and shape. And even more, shape really isn't a factor because when we're talking about shape, we're talking about how proteins coil and how they fold up, but we're unfolding our proteins. So you can really just think of SDS page essentially as filtering proteins by size. And that's a very high yield concept that is almost guaranteed to come up on the MCAT. And so again, just to really solidify this concept, since we're only filtering by size, the molecular weight of our proteins, or their size, is what's going to determine how far they travel on a gel. And so the way that this is helpful is we know that because of the image I showed you on the last slide, smaller proteins will travel further and larger proteins will travel not as far down a gel. And so what you can do, or what scientists do, is they insert not only the sample, but they also insert a reference ladder with known standards in it. So they know that, for example, this protein is 100 kilodaltons, this one's 50, and this one's 25. And then so by comparing across your gel, you know that, oh, the smaller protein we isolated is around 25. The larger one's around 100. And so this is the way that scientists are able to study the physical property of size of their proteins of interest. And so now that we've thought about how we can separate proteins from each other based on size or charge or shape, well, you might be wondering, how do we even get those samples in the first place? How do we even separate the proteins from everything else in the cell that we've harvested? And again, it's the same idea. We're going to be exploiting the physical properties of whatever we're trying to purify. And so these separation techniques that I'm about to present to you are very high yield. And if you think about the logic behind them, are usually pretty simple to understand. You just need to think about them in terms of exploiting the physical property of something, usually a protein, in relation to whatever else it's mixed in with, in order to isolate it, in order to get a pure sample. And so let's do that. Let's think about that. The first one we're going to be talking about is size-exclusion chromatography, also known as gel filtration. And this is going to be, again, utilizing the physical property of size to isolate proteins. And so our experimental setup is we're going to have a sample up here, as you can see, mixed with multiple components, some sort of column, some sort of porous beads within the column, and then some sort of container for whatever we're isolating from this column. And so you'll notice that inside this column, we have porous beads, so they have small holes within them. And so thinking about the perspective from a large protein, we're going to see that it's going to travel down, 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 not get stuck in these small beads and end up eluting first into our column. However, a smaller protein is going to do the same thing, travel down, 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 and then whoops, it's going to end up in one of those smaller holes and get stuck inside or adhere to our beads. And so you can see in this manner, by using these porous beads within our size-exclusion chromatography experiment, we're able to allow bigger molecules to elute first, and then smaller molecules to elute second or elute later. And so one way the MCAT might try to trick you is you might see the words size-exclusion chromatography, you might think of beads in a column, and you might say, oh, it's obvious, the smaller beads are going to elute first, the bigger beads are going to travel slower, or the bigger proteins are going to travel slower and elute second. Well, that's not the case. These beads have small pores, and it's specifically meant to trap smaller proteins and allow bigger ones to elute first. In addition, we also have ion exchange chromatography, and the physical property we're going to be exploiting for this technique is charge. And so you can see, very similar experimental setup. We have some sort of mixed sample, we have a column, we have beads, and we have some place for our sample to elute once purified. The only difference is now instead of porous beads like we had over here, we're going to have charged beads, and in this case, negatively charged beads. And so you might be assuming, or you might already know what happens just by looking at this experiment. If you have a positively charged bead, or positively charged protein, it's going to be attracted to those beads. It's going to want to stay in the column because it's surrounded by negative charges, and that creates favorable interactions. However, a negatively charged protein is going to want to go through the column really quick because all these negative charges are going to be repelling it and squeezing it through that column. And so you can see that this is a pretty simple way, pretty simple experimental setup to separate proteins with positive and negative charges from each other. And so one thing to keep in mind during these experiments that might not be clear from the experimental setup is that there's often an elution step, or what I like to think of as a rinse step. And so what this step is essentially you're going to end up with, in this case, negatively charged beads with positively charged proteins attached, but you want those proteins to come off eventually. Maybe not at the same time as the negatively charged proteins, but eventually you want them off. And so what you do is you rinse the beads and the proteins, and in fact the whole column with some sort of solution with a salt or pH gradient. And so what this is going to do is it's going to take advantage of the PI we were talking about earlier to make the proteins negative or neutrally charged, so have no charge, and then they're just going to fall through the column in a elute. And so by controlling when this new solution is added to the column, you can control when you're eluting negatively charged proteins. Then you can add a new container, for example, and then control the elution of the positively charged proteins. And so this really allows you to effectively and efficiently separate proteins with positive and negative charges from each other. Okay, and so the next separation technique we're going to be talking about is isoelectric focusing. And like all the other techniques we've talked about so far, this one is going to be utilizing a physical property of peptides to separate them, which in this case is going to be pI. And so we talked about pI earlier. We know that pI is the pH at which our peptides or our protein has a net charge of 0. It is a Swither ion. And so let's look at how this experimental setup utilizes this physical property to separate peptides. So let's say we have a gel or a membrane, which I've drawn out. And on one side of the gel, we have a high pH, and on one side, we have a low pH. And everywhere in the middle, we have pHs of varying magnitude. And so we've established as experimenters a pH gradient. In addition to the pH gradient, we also have an electrical current running through our membrane. And so let's think about what's going to happen. Let's think about, from the perspective of a protein, how this experiment works. So let's say we have a polypeptide here. And let's say this polypeptide has a pI around 7. And then let's say the pH right here is 7. We know that if we place our peptide down here at this end of the gel, because of the pI being greater than the pH at this point in the gel, we know that our peptide is going to be charged. And even more, we know that our peptide, in this case, is going to be positively charged. And so because of the electrical gradient, as well as the pH gradient, our peptide is now going to migrate through the gel until it reaches the pH of 7, where at this point, the peptide will be a Zwither ion and have a net charge of 0. And so by studying the migration of different peptides and by studying where they end up on the pH gradient, we can separate proteins by their pI. And so if you know that your protein of interest has a specific pI that none of the other proteins in your sample have, this is the method you would use to separate it because it efficiently separates your proteins based on their specific physical property of pi. Additionally, another method we're going to be talking about is affinity chromatography. And this uses the physical property of affinity, or binding affinity, to separate peptides. And one reason why this method is so important and why it's most likely to come up on the MCAT is because it is very highly specific. So this method will efficiently and effectively separate proteins in a very highly specific manner. And so the way this works is by utilizing antibodies, which you can sort of think of for now as these Y-shaped proteins. And so we will talk about that later when we go into the immune system, but essentially for now, we can just think of antibodies as binding specific proteins. And so only proteins that match the variable regions of the antibody will be able to bind them. And proteins that don't match that region will not be able to bind them. And so, in general, these antibodies typically have affinity for only one molecule. And even if it's one or two molecules, they are extremely similar in structure and extremely similar in peptide composition. And so, thinking about this, that's how this method works. You can place, like the other experiments, different beads within an elution column. And attached to the beads, you can add antibodies that are specific for one peptide. And so, when you're eluting, a different sample with mixed peptides through the solution, every peptide that doesn't bind to the antibodies is simply going to flow through them and end up eluting first. Meanwhile, your protein of interest is going to bind to the antibodies and it's going to get stuck in the elution column. And so you might be wondering, okay, cool, we've taken out everything we don't want, but now all our proteins of interest are stuck in the elution column. How do we get them out? Well, similar to ion exchange chromatography, we can now pump in a solution that unbinds our protein of interest from the antibodies. And then we can add a separate container to the bottom of our column, which collects these protein of interest. And so these solutions are usually utilized in what's called a wash step. And they can even utilize substrates with even higher affinities for the antibodies that then comes in and kicks out these proteins of interest, which then makes them fall off or unbind the antibodies, elute through the column, and end up in a new container, which we've placed at the bottom of our elution column. And the last separation technique we're going to be talking about is centrifugation. And this separation technique, like the others, utilizes a physical property to separate different cellular components. And in this case, it's going to be the density of different peptides in this example, but it also can be nucleic acids or other cellular components. And so how this technique works is essentially you have different containers with mixed proteins or mixed cellular components, all of different sizes and densities, all mixed together. And what you do is you then place these containers inside a centrifugation machine, like the one I've shown here through this diagram. And what this machine does is it essentially spins this capsule with all your samples in it at maybe 10,000 or even higher rotations per minute. And so it just spins it really, 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 really fast. And the way that density is utilized in this experiment is that because of density and because of buoyant forces, all our smaller molecules will migrate towards the top of the sample, and all our heavier, more dense macromolecular components will migrate towards the bottom of our sample. And so after spinning these samples at 10,000, 100,000 RPM for a few minutes, you then take out your Eppendorf tube or this container with your sample, and you'll notice that at the top you'll have a solution with all your smaller, less dense macromolecular components, and on the bottom you're going to have those more dense components, usually formed into a pellet or an aggregate. And so by being able to understand cellular structure and the different density of different components, you can use centrifugation to isolate whatever your molecule of interest is. Because you know that proteins, which are usually smaller, are going to end up in this solution layer up top. And then, your cell wall, your nucleic acids, all those huge macromolecules are going to end up in the bottom. And so that's it. That's the last separation technique I've prepared for you. And if you can just be able to understand the fundamentals or the basic experimental logic behind these techniques and behind PI, you should have a much easier time on the biochem section of the MCAT because these topics are most likely to appear. They are very high yield. And that's it. That's all I have prepared for today. So good luck with your studying, and if you have any questions, please feel free to leave them in the comments below.